Hey, what's up everyone, and welcome back to the Sanctuary. I'm your host, Professor C, and today we're going to do a little bit more A and P. Specifically, we're going to talk about the second portion of the central dogma, which should be transcription of DNA. So, let's get to it. Alright, so for the second part of our discussion on the central dogma, we'll be talking about transcription. The one that I think is the easiest of the three: replication, transcription, translation. And remember, the focus of this talk is RNA, RNA, RNA. Even though this is DNA to RNA, DNA will be the minor player here. So looking back here at our central dogma picture, we've dealt with replication in the previous talk. But I like to put a wall here when I talk about the central dogma a little bit, and just so you can physically see that we're moving into the other side of the wall. Remember, with replication, we copied all of the DNA, and remember we did this prior to cell division so we can give a full set of info to our offspring cell. On the other side of the wall here, we're actually going to go from DNA to protein. We're actually going to cook up some protein now. Finally, we're going to read some recipes, cook some stuff up, get some amino acids, chain them together, and make a protein. But you have to go through a middleman called RNA. And that's what the central dogma tells us. You can replicate DNA all day long, just like we looked at last time, but you'll never get protein out of it. So we have to switch courses here and try to get some protein from our information called DNA. And it's a two-step process. We're going to do transcription, which I often write as TC when I'm doing notes real fast, and translation, TL. We'll do transcription this talk and translation the next talk, but it's a two-step process. We cannot just go from DNA to protein. I will make a note down here. There is this thing called reverse transcription. You could go from RNA and back copy to DNA. So some viruses do this, especially those that are called retro. Again, retro meaning backwards. A retrovirus would do this. It would work backwards, and it would make DNA from RNA. All right, so let's look at the process today of transcription. The main difference here between replication and transcription is that we're only going to copy only one gene. We're only going to copy a small piece of the DNA here. In fact, it's just one recipe, right? The gene is the recipe. We're only going to copy one thing. So we're going to cook some food tonight, but we don't want to cook every recipe in our cookbooks, right? We just want to make one thing. All right, RNA, as we said before, it kind of looks like half of a DNA. We need to talk about specifically three types. We may have touched on this when we talked about nucleic acids a long time ago during the chemistry section, but let's do it again in case we've forgotten. So three types of RNA are found in your cells. All of them are needed to do protein synthesis. If we don't have one of them, we can't do anything. rRNA is the thing that makes up the subunits. Remember that a ribosome actually looks like a big, to me it looks like hamburger buns. They have a big hamburger bun on top of the small bun. And we call this in another talk, we said this was the 40S subunit and this was the 60S subunit, or we could just say that's the small and the large subunit. So what are those two subunits made of? rRNA. And if we remember our organelle talk, that rRNA is concentrated in the nucleolus, which is buried inside the nucleus. So we need some ribosomes. If we're trying to think of, an analogy to make this simple, I'm going to call the rRNA, the ribosomes, they're going to be the ovens where we're going to actually cook up our cake. Remember, we're trying to make a protein here. The rRNA will serve as the place where we cook it up. tRNA. tRNA looks a little different. tRNA will kind of have some arms sticking out here, a big head, another arm sticking out here, and then down below it has 3 teeth hanging off. And this is a pretty good picture. Not only does it look kind of like a T, it's kind of like a cross, but that's not why it's called tRNA. It's called T because it's transfer RNA. And so hanging off of this, you should find, and I'm just going to put it simply AA, one of the 20 amino acids will be hanging off of it. So its job, a transfer RNA, is to transfer. Transfer what? Amino acids. Remember, if you want to cook up a protein, you got to have the ingredients, right? So sometimes I call the tRNA the chef. He's the guy that's got to bring the ingredients over to the oven. So bring it from where? Well, from the cytoplasm to the ribosome. That's what the chef will do. That's what the tRNA will do. Take that amino acid and bring it from the cytoplasm to the ribosome. We need a third player, mRNA. mRNA looks like a ribbon. And it's what you see on the very first image of this talk is a ribbon-like, kind of like half of a DNA, right? It's got ribose in the backbones. All of these do. It's got U's. All these have U's instead of T's. What it is the copy of the gene. And to keep the cooking analogy here, this is the recipe. It's not the actual DNA recipe. The actual DNA recipe is going to be kept safe in the safe. But we want a copy of it. Just like if you had grandma's cookbook and you wanted to get a recipe for whatever, you're probably not going to be able to tear that out of grandma's cookbook. She probably wouldn't like that. So you can't tear the information from the DNA, but you can copy it, right? You can copy down grandma's recipe and then read it later and cook it up at your own house. That's the concept. So 3 RNAs, rRNAs being the ovens, tRNAs being the chefs bringing the ingredients to the ovens, and the mRNA, the messenger, being the actual copy of the gene. Remember, we're only going to copy one gene here in transcription. It is the recipe. It is the copy of the gene. If you're ever asked where RNA comes from, in a modern cell, it's very clear. All RNA is transcribed, again, through the process of transcription, which we're about to see in more detail, using an enzyme we've seen before. If you watched the talk on replication, you saw RNA polymerase is an enzyme that lays down ribonucleotides, and thank goodness that's what we want to do here. We want to make nothing but RNA, so we want somebody that lays down only ribonucleotides. All RNA in modern cells is transcribed from DNA by the same enzyme, RNA poly. So RNA poly will be the enzyme. It will transcribe that mRNA from a strand of DNA. DNA, a gene, will be a template. And if you want to say from a coding strand of DNA, you could say that's, a gene. So RNA poly will copy a gene. And just to remind you, this is a big sticking point when I taught this in the classroom. Unlike replication, in which we copy the entire molecule of DNA, here we're only going to copy a small part of it, only one gene. Again, you don't want to cook up everything in grandma's cookbook tonight. You just want to cook up one specific thing, one gene. One protein is all we need to make. New strand, single-stranded, RNA, made of ribonucleotides, not deoxyribo. It's got ribose in the backbone like it's supposed to. It's got uracil instead of T's like it's supposed to. Okay, let's draw a little sketch of what's going to happen here. So I'm going to have some DNA, and I'm just going to make this like that. Now you're saying that doesn't look like DNA, it looks like an RNA. Well, the other half is down somewhere else, right? It's already been split apart in a fork, and we're going to have a fork here too. We just kind of skip over that part. But remember, we just need to copy one gene here, and I'm going to say this is the gene. Now the DNA will continue on and on and on and on and on and on and on. And you can kind of see that here. If you have an entire DNA molecule, before we opened it up, we could say, gene is just, from here to here. There's the gene. Just a small part of that DNA. That's what we're interested in copying. If I could see it like down here, when it's all opened up, it's the same thing. I've taken that gene and I've just isolated it so we can see it. So what we're going to do is we're going to have a section in front of the gene called a promoter, and a section after the gene called a terminator. And all these words, if you hear promoter, terminator, or gene, you're going to think DNA. And that's what we're going to copy, is a single strand of DNA will be copied into a single strand of RNA. So let's bring up some of the text here. So there's 3 major events of transcription. I've put them in pink, and I've put some of the other keywords here in other colors. So initiation is step 1. And it begins when some sort of RNA polymerase, RNA poly, binds to a promoter, a short sequence of DNA upstream from a gene. Simply, I just take an enzyme called RNA polymerase, and plop it down on the promoter. And there we have initiation of transcription. If we could fast forward, right? I can't show it as an animation, unfortunately, but I can do this. I can fast forward and pause. And I can tell now that my RNA poly has started scooting along. Again, it's still going to roll 5' to 3', but we're not so interested in that because we don't care about the other side of the fork here. There's no leading lagging strand in transcription. That's only in replication. So our poly's running that way toward the terminator, and this phase is called elongation. Elongation. And what's happening as poly moves, it says she's adding ribonucleotides using complementary base pairing. So let's pretend this is G, C, T, because it's DNA, T, C, A, C, T. Let's just pretend. Poly is copying that into a RNA form, and it's going to be coming out like a tail. It's not going to be stuck to the DNA. So here would have an A, right? And a G, and then a U would be the next base, and then a G, and then A, G, C. All I did was work backwards and base pair complementary. She's adding ribose in the backbone. She's adding, obviously, uracil instead of thymine. So this will continue and continue. If poly scoots forward even more, and I'll just kind of show you the example here. If poly's all the way here now, still scooting, what's going to be the tail look like? It's going to be longer now, right? It's going to be longer. That's why it's called elongation, because we're making the tail longer. This is RNA that is coming out the back end. All right, step 3 is very simply termination. When poly gets to the terminator, there at the end of the gene, everything stops. It releases, everybody goes home. All right, let's take another peek here. Let's transcribe a gene. Step 1 would be initiation. And to do initiation, what do? You take an RNA poly. I'll just put R right now. It's an RNA poly, and we plopped it on the promoter. And we're done. The next step is called elongation. In elongation, the poly starts to slide along. So let's pretend poly's here, sliding along the gene, 5' to 3' toward the terminator. What should be happening? Well, there should be a tail coming out of here with these bases that are complementary to the bases on the gene. So again, you can do this more if you want to stay and practice it a little bit, but G and then this T goes with an A, and then a C, then an A, then a G, then a C, and I'm just base pairing, except you got to put U's in here whenever you try to put a T, A, and so on and so on and so on. This is called elongation. And of course, at the very end of it, when you hit the terminator, everybody goes home. The RNA falls off. The polymerase goes and does another job, as enzymes are wont to do. And the DNA, the gene, usually closes back up. But there's the concept of transcription from promoter to terminator. Pretty straightforward. Thanks for watching the video. If you liked it, check out some more. And I will see you on the next one. One more to go with the Central Dogma. Bye-bye.